Hi everyone, today we're going to look at a very important hormone which is insulin, and we're going to talk about the synthesis and mechanism of release of insulin. So first of all, we're going to deal with the synthesis of insulin, then we're going to look at the insulin receptor and what are the cellular changes that it brings about. Then we're going to talk about the mechanism of release of insulin, and we're going to look at two graphs right here. So first of all, the synthesis of insulin. Insulin is a very important hormone which is released by pancreas. And what region of pancreas specifically? The islets of Langerhans. And islets of Langerhans has a variety of cells. It has alpha cells, beta cells, and it has delta cells. The alpha cells, they have the role of secreting glucagon. Beta cells secretes insulin and amylin. Then the delta cells secrete somatostatin. Then you have P cells as well, the PP cells, which secrete pancreatic polypeptide. But right now, we'll be primarily focused on beta cells because they secrete insulin. And what else they secrete as well? They also secrete a myelin, but our major focus is insulin. Now, the site of synthesis of insulin that we've already understood, it is pancreas. Now, pancreas, they have two kinds of cells. They have acini and they have islets of Langerhans. When you talk about islets of Langerhans, I've already told you the type of cells that they have: alpha, beta, gamma, and polypancreatic pancreatic polypeptide cells, which is the PP cells. So these are the four types of cells that they have. So this has endocrine nature. It secretes the hormones, insulin, glucagon, and all of that. What about acini? Acini is responsible for secreting the digestive juices. So digestive juices are very important for digestion. So it has an exocrine nature as well as an endocrine nature. This is the role of pancreas. Now, talking about the synthesis of insulin, let me give you a rough overview. So, first of all, you have pre-proinsulin that gets transformed to proinsulin. Then you get insulin, which is packaged finally into a vesicle and it is stored. Um, what are the steps? Starting off with the gene transcription. First of all, what happens in a transcription? Basically, a gene is transcribed into mRNA. So forming an mRNA strand, that is transcription. Then translation happens of that mRNA strand into a protein. What protein is that? That is pre-proinsulin. Now pre-proinsulin, it is made up of a few components. Let's discuss those. First of all, it has two alpha. Okay, let me just change the color of the pen. It has two alpha subunits. So these are the alpha chains. It has two alpha subunits, then it has two beta subunits. Let's draw those as well. It has two beta subunits. It has a C peptide chain. Its role is to connect the two. And it also has a signal peptide. It also has a signal peptide. So this is what? This is our very important molecule of pre-proinsulin. Pre-proinsulin. What are the components again? It has two alpha subunits, it has two beta subunits, it has a C chain or C peptide, and it has a signal peptide. Now, this is pre-proinsulin. And where is it formed? It's formed primarily in the endoplasmic reticulum, the rough endoplasmic reticulum. Then moving forward, this pre-proinsulin, it gets converted to proinsulin again in the rough endoplasmic reticulum, where what happens is that the signal peptide, it is removed. So the cleavage of the signal peptide occurs and you get the proinsulin. So now what does the proinsulin comprise of? Since we've removed signal peptide, so proinsulin has two alpha subunits, two beta subunits, and one C peptide. Let's look at this diagram, which is from the book Guyton. So this is the proinsulin. So pre-proinsulin got converted to proinsulin. What does the proinsulin have? It has alpha chain, it has beta chain, and it has a C chain, right? So yes. And now moving forward, you have there's another very important thing. Okay, did I make any mistake about the number of chains? Because here we can only see one chain. Um, two amino acid chains. So there should be only one alpha and one beta. I've made a mistake. Sorry. There is one alpha and one beta. So this is a wrong diagram. Sorry. There should be one alpha and one beta. So let me just remove this. Yep. And let me alter this diagram. There's one beta chain and there's one alpha chain. Is this the same color? It's not. Which color is it? Is it this or is it this? Yes. So one alpha, so it has two amino acid chains. What does it have? It has two amino acid chains, and those amino acid chains are one alpha and one beta. So this is something that you need to know, and I'm sorry for the mistake. So it has two amino acid chains, one alpha, one beta, a C peptide, and a single signal peptide. Signal peptide is removed, and that's when pre-proinsulin gets converted to proinsulin. And this happens where in the rough endoplasmic reticulum. In rough endoplasmic reticulum, the signal peptide is removed, and the proinsulin it folds, and disulfide bonds they form between the alpha and beta subunit. So this is the proinsulin that you can see right here. A chain, the alpha chain, the beta chain, and there's a C peptide chain, which is the connecting chain. Then what happens? This proinsulin gets converted to insulin. Where does this happen? This takes place in Golgi apparatus, and in this, what happens is that the C peptide that is removed. So initially, in order to form proinsulin, in from pre-proinsulin, we had to remove the signal peptide, and then when we were forming proinsulin, we had to remove when we had to form insulin finally from the proinsulin, we had to remove the C peptide chain, and we finally got the C peptide. I hope you're understanding what I'm saying. Let me draw a flow chart here so that you understand better. This is pre-proinsulin, the molecule that I drew above. This pre-proinsulin, this one, it got converted into proinsulin, and what happened? What was the change that took place here? We cleaved or we removed our signal peptide. So you got the proinsulin. And then you want the insulin. In order to form insulin, you have to remove what? You have to remove the C peptide. And finally, you got insulin. Now, what does insulin have? It has one alpha chain, one beta chain, and a lot of disulfide linkages in between them. So here you can see C-peptide is removed. You have insulin, which has alpha and beta subunits with disulfide linkages. And this step took place where? In Golgi apparatus. This happened in the rough endoplasmic reticulum. This also, and even the first step happened in the what do you call it? Rough endoplasmic reticulum. This step took place in the Golgi apparatus. Now, insulin gets stored in the granules as zinc insulin hexamers, and C peptide is also stored within the same, uh, and it is released in equimolar amounts. It's stored within the same vesicle, and it's also released in the equimolar amount. So, this is the synthesis of insulin. Now, moving forward, let's talk about this very important step, which is about the reception, where your insulin molecule will come and it will bind to the beta cells. But for now, we made the insulin, right? Insulin has been made. But before talking about this, we need to discuss the secretion, because once secretion happens, then only the insulin molecule will come here. So right now we were in the pancreas where we made our insulin. Now let's talk about the insulin release from the pancreas. Insulin release from the pancreas. So what happens is that first of all, uh, you have this glucose transporters, right? And how do these glucose transporters come up here? That's also a long story where the vesicles of the glutes they basically get translocated, and then you get a glucose transporter here. Through this glucose transporter, glucose will come in. This glucose will be phosphorylated to glucose 6 phosphate with the help of glucokinase. This is the rate limiting step, right? And then, or the rate determining step, rate limiting step, and then this glucose 6 phosphate, its oxidation occurs, and we ultimately get ATP. So, getting ATP is the ultimate step. Why? Because ATP is going to do what? It's going to block this ATP and potassium channel, ATP based potassium channel. ATP sensitive potassium channel. This will be inhibited. So when this gets inhibited, what happens is that there won't be potassium. Now there'll be depolarization. And why will be there depolarization? So the answer is very simple, because if there's sodium channels, because of influx of sodium, you will get depolarization, and because of efflux of potassium, potassium efflux gives you repolarization. But imagine if the potassium channels are closed, there won't be efflux and there won't be repolarization, so you'll just have depolarization. And along with the closure of these potassium channels, there will be opening of the calcium channels, and calcium goes in. And what will it do? The influx of calcium is going to stimulate the fusion of the docked insulin containing vesicles. These vesicles are docked here; they're just waiting here patiently. So what will happen? They will be translocated and they'll fuse with the membrane, and there'll be exocytosis of insulin, and that's how insulin is released. So this is the entire mechanism. And what does ATP do? ATP has a very important role. It inhibits the ATP potassium channel. This is one thing that you need to know, right? Because this entire procedure is very easy. Just you need to learn this part, that ATP inhibits the ATP sensitive potassium channels, because of which depolarization occurs, potassium channels open, and fusion of these vesicles with the membrane, and insulin is released out. Then this was pancreas. Now pancreas released, they synthesized insulin, and pancreas released insulin. Now this insulin is going to go and act on the muscle and adipose tissue cells, adipose cells. What will happen? Insulin will come and it will bind to this alpha. There are two alpha subunits and there are two beta subunits. It's going to bind to this insulin receptor. This is alpha 2 beta 2 heterotetramer receptor, which is also known as enzyme linked receptor, because it is linked to an enzyme internally. I've already made an entire video on this, so you can go and look at that if you want, but I'll just do a quick recap. So it's going to activate this target receptor, which is on the muscle and adipose cells. And because insulin binds here on the alpha subunit, but alpha subunit is connected with the help of disulfide linkages to the beta subunit, there'll be autophosphorylation of beta subunit. This will activate the tyrosine kinase, which will cause the phosphorylation of insulin receptor substrates, which are the intracellular receptors. And they are going to mediate the effects of protein metabolism, glucose metabolism, fat metabolism, and also these vesicles will be forced to translocate, and the glutes will be released and they'll be integrated into the membrane. So, yes, so this is the effect of the insulin, what happens afterwards, and the intracellular effects. The four primary intracellular effects are that muscle cells and adipose cells will be increased uptake of glucose. More and more glucose will come into these cells via these glutes, and they'll become more permeable to amino acid, potassium, and phosphate ions. There'll be intracellular metabolic enzyme. They'll be either activated or either they'll be deactivated. For example, it activates these, you can also deactivate them, and then there'll be changed rates of translation of mRNA. Now moving forward, when we're talking about the mechanism of insulin secretion, there was a certain paragraph that needs to be discussed, and that's written in Guyton. You can refer to chapter 79. So let's read every line word by word and then understand it. Other nutrients, such as certain amino acids, can also be metabolized by beta cells to intracellular ATP levels. Other nutrients, such as certain amino acids, can also be metabolized by the beta cells to increase intracellular ATP levels and stimulate insulin secretion. What are they saying? They're saying that here I told you that glucose came in and then we got ATP, and then ATP inhibited this, right? So they're saying not just glucose, you can also have amino acids. So that's a good thing that we got to know now. So let me just write this, let me just jot this down somewhere. So first we spoke about glucose coming in and getting us ATP, because glucose got phosphorylated, glucose 6 phosphate, then glucose 6 phosphate oxidized, and we got ATP. But not just glucose, even amino acid can do this role. So that's the second thing that we discovered after glucose. The second line of this book said some hormones such as glucagon like peptide 1, glucose dependent insulinotropic peptide, and acetylcholine, they also increase intracellular calcium levels through other signaling pathways and enhance the effect of glucose. So they're saying that there are these substances, which I've already mentioned, including acetylcholine, they increase intracellular calcium level. So yeah, ultimately that's one thing we need. We need increased calcium levels so that they can go and they can cause the fusion of these dogged intracellular vesicles that contain the insulin, so they can cause a fusion of these vesicles. So it's a good thing if we are directly, with the help of all of these substances, we are directly having influx of calcium, and that is causing the exact same effect that otherwise we would have gotten if we followed the entire pathway. So this is the third thing that we're learning, that there's some hormones that can also do these roles. And the hormones names I've already told you, they're quite tricky. They're quite tricky, but yeah, you can know them if you want. Moving forward, certain hormones and neurotransmitters do not directly increase ATP. Instead, they act through cell signaling pathways. These pathways cause increased intracellular calcium in beta cells. Calcium is the final trigger for insulin release. These substances enhance glucose stimulated insulin secretion. So, they basically enhance glucose stimulated insulin secretion. Although they do not have major effects on insulin secretion in the absence of glucose. These hormones cannot cause significant insulin release alone. Glucose must be present. That's the reason they have this glucose dependence. So glucose has to be there. It's not that, oh, there's no glucose, so this entire pathway is not required and this is happening. This will happen directly also, but for that you need at least some amount of glucose. So that some amount of glucose will be causing this, but there'll be some hormones which will also be causing this, and they'll be taking a shortcut. So you ultimately you do need some glucose for even for the third step, this third thing to happen, the hormones thing. For amino acid, you do not need the glucose, obviously. Other hormones including somatostatin, so this should be highlighted in blue, and norepinephrine, they do what? They activate alpha adrenergic receptors. So they inhibit exocytosis of insulin. So this is important for you to know that somatostatin and norepinephrine. So that's the reason they mentioned here. Somatostatin and alpha adrenergic activity, which is caused by the norepinephrine. So these are going to inhibit the insulin secretion. That's the reason they are in this domain. And moving forward, the next line says some hormones reduce insulin secretion. Somatostatin directly inhibits beta cells. Norepinephrine activity via alpha adrenergic receptor suppresses insulin release. So what are they affecting? They're blocking the exocytosis. That's the reason insulin secretion is blocked. And then, very important, we have, okay, so I think I think I should also mention this thing here, but this should be in the inhibitory side of it. Somatostatin and norepinephrine, which works with the help of alpha adrenergic receptors. This basically inhibits the entire exocytosis. Moving forward, they're saying that sulfonylurea stimulates insulin secretion by binding to ATP sensitive potassium channels and blocking their activity. So very important. These are anti-diabetic drugs. What do? They do a very good role that they directly bind to this. So they bind to the ATP sensitive potassium channel and they inhibited sulfonylurea drugs. And once these are inhibited, the potassium channels will open, the fusion will happen, insulin will be secreted, right? And insulin will cause glucose uptake. So these are anti-diabetic drugs. So sulfonylurea, they do the same function as the rest of the things they're doing. Sulfonylurea. Then you have here very important. Moving forward, these mechanisms results in the depolarizing effect that triggers insulin secretion, making these drugs useful in stimulating insulin secretion in patients with type 2 diabetes. So these were the things that we studied, and let's recap what were the four things. Glucose, it follows the entire step. Amino acid, it also follows the entire thing. What about the hormones? Hormones, they use some other signaling pathways, and because of that in other signaling pathways, it directly increases the calcium influx. Somatostatin and norepinephrine, they inhibit the exocytosis part of it. Sulfonylurea, it does what? It directly inhibits this, and because of which there will be influx of calcium. And here you can see increased blood glucose, increased blood free fatty acid, increased blood amino acid, and then there's some hormones which we already discussed. What do they do? They increase the calcium influx directly. And then we had sulfonylurea drugs. What do they do? These are the examples, and these directly block the potassium channels. Then moving forward, this is the graph that tells you that these are the an increase in plasma insulin concentration after a sudden increase in blood glucose. So if a person has consumed a lot of sugar, let's suppose, or any food, as a matter of fact, after after a few minutes, the insulin activity or insulin release will be increased a lot. But then it will simultaneously decrease as well. It will also not simultaneously, but after a few seconds, it will decrease, and then there'll be a further increase in insulin secretion, and that will be uh, so there'll be, first of all, there's an initial rapid surge, and then there is a delayed but higher and continuing increase. So this is delayed, late, but higher, and it's constantly continuing increase in the concentration. So let's talk about these two. So the initial rapid surge is because of immediate dumping of the preformed insulin. So there's preformed insulin that is immediately dumped out of the beta cells of islets of Langerhans. That's the reason for the initial high secretion, but it's not maintained because obviously those that already preformed insulin store, it obviously gets depleted. And then the second increase, the plateau, that is because of uh, the preformed insulin as well as activation of enzyme system that produces insulin. So there's an enzyme system that is going to produce insulin. That enzyme system will be enhanced and there'll be a lot more insulin. So that this is going to take time. That's why it is in a second plateau. Now, moving forward, this is another graph which shows you that normal levels of glucose concentration in a person should be 80 to 90. Beyond 80 to 90, let's suppose beyond 100, there'll be a rapid increase in insulin secretion. So our body is like very closely monitoring the glucose levels. So glucose is a stimulus and it causes increase in insulin, and the increase in insulin is both dramatic and it's also very rapid. And the rise in glucose increases insulin secretion. And what does the insulin do? It increases the rate of transport of glucose into liver, muscle, and adipose cells as well. So, yeah, that's pretty much it. This was a quite extensive video, but I'm sure now you're clear about the synthesis and mechanism of release of insulin and also the reception of insulin by insulin receptors, which are enzyme linked receptors, tyrosine kinase type.